All right. Then I want to do full screen. Oh, but that is terrible. My face does not need to be that big. Y'all want to hit the lights for me? Y'all want somebody over there to get the lights? Thank you. Yeah, we could do that. We could do that.
I'm okay with like what? Can we have nap time for class today?
No, we may not have nap time for class today.
Oh. You're
Good
about to show us
try.
a video, aren't you?
Am I to what?
Are you about to show us a video?
No.
Then why is it so hard?
Reclaiming host. Okay, that's whatever.
What's it called? These
It's not video.
daylight.
Are we just taking naps today?
That's what I said. What, nap time? Can we, these have the nap time for class instead of white light?
That's fine. Can you still see the screen? Is the only question. But yeah.
I can see it's just fine.
All right. Look, I'm not too far behind.
Huh?
So I'm not too far behind. It's 9:01. All right. Oh wow, that's a huge glare on the screen, but on this of the screen on this for me. But it's fine. As long as y'all can see, it's cool. All right. So I do not have your test graded yet. It's gonna take me a little while.
Just to get these next Tuesday. Before I start asking.
I'm gonna do my best, but just be
It's
patient.
all discussion.
It's all discussion. It takes time.
Yeah.
The other thing is we might be virtual on Tuesday.
Oh,
Charles might have a doctor's appointment, so
Mike.
might. We will see. So I'm just giving you a forewarning.
I'm gonna cancel that.
Okay, so it's okay. Well, I didn't check that. I need to go check that. See who else is on my schedule. It's a good thing. Okay, thank you. What else? I feel like there was something else I wanted to show you. But I'll tell you a joke. I'm sure you know this one. Did I tell you the one? Did I tell you one about not telling secrets on the farm?
No.
Oh, okay. So why shouldn't you tell secrets on the farm? Because the corn has ears, the potatoes have eyes, and the beans talk. The beans talk. I like the bean talk. Okay. And then this was the other good one from today. What did the beach say when the tide came in? Wait, I've forgotten the punchline. No. It's nice. It's been a while since I've seen you. No, how does it go? See, it's about the sea. It's nice to see you. No, but it's something else. It's something else. Oh my God, that's terrible. Something about see ya. Oh man. This is why Charles carries around the card and I don't get the card. Should take a picture of it with my phone. That way I get it right. All right. So we are talking. Whoops. Amino acids. We're moving on. I feel like we're getting close to the end. When we get to amino acids, I feel like we're really close to the end. All right. So this is full screen. Okay. All right, so when we talk about amino acid metabolism, I kind of want to take a side trip before we start taking notes and stuff. So do y'all know what Roundup is?
Yes, pesticide.
It's a pesticide, right? Okay, so how can you spray pesticides and kill plants you don't want and keep plants you want? How does that work? Genetic modification. Right, so there's a big company that has the rights to Roundup. Right, y'all know about this a little bit. So why is it? What's the compound? So inside of Roundup, if you want to know this, Roundup is glyphosate. So glyphosate. So this is the active ingredient in Roundup. So the thing with this is it inhibits the synthase, a synthase enzyme in the metabolism or the building up of aromatic amino acids. Okay, so it inhibits all aromatic amino acid synthesis. Okay, so if I spray this, no matter where I spray this, every natural plant that it hits dies, right? So now I'm going to create another side market where I'm going to genetically engineer crops. And I'm going to give those genetically engineered crops an extra gene. So I am going to give them a bacterial gene for the same synthase enzyme, right? So I give plants a bacterial synthase, which is EPSP, and it's a really long name, but a bacterial synthase gene that is resistant to my drug, right? So now what happens? If I spray the whole field, what happens? As long as every single plant that I plant is genetically modified, the weeds die and the GMO lives, right? So you can increase by about 20% your yield from any given field using something like this, using a mechanism like this. 20% is huge. 20% is huge. So you can make a big difference in the amount of crop that you can get on a certain plot of land, which is really important for eventually one day, hopefully, curing world hunger, right? Eventually, hopefully. But are there no side effects? Excuse me. What kind of side effects? What do we have to worry about? What are you doing to the native plants? What if this gets into the runoff? How are you affecting the local ecology? What's that? It can. You have to worry about drainage. So you really have to worry about how you design your field. What else? What else could be a problem? Well, we don't synthesize them. So we have to eat the amino acids. So that's okay. Eating the plants with but they're dead if they've, oh, you mean like the crops, the actual crops. Usually go through a washing period, so that's usually not a problem. What happens, right, when evolution finds a way, like it always does, and you get weeds that are now resistant to this Roundup? Now you're gonna not have anything to treat these entire fields, and then the weeds are going to insanely start to take over, right? So is there a perfect answer? No. But is GMO in and of itself a bad thing? No, but you have to worry about how it affects the economy. You have to worry about the off-target effects that you might not plan to have, right? So when you think about all that, just because it's GMO or just because it's non-GMO doesn't mean that it's good or bad. Okay. All right. So why do we talk about all that stuff, right? So all of that has to do with inhibition of what? Amino acid synthesis. So we're going to talk about amino acid metabolism. So the first section, we're going to talk about nitrogen fixation. So what is nitrogen fixation? Atmosphere to an organism, right? Atmosphere to organism. So that's fixation and assimilation. Then we'll talk about how we break it down, how we build it up, and then what else can we make from amino acids? Yeah, we're going to talk about, there's lots. There's lots. It's very, very interconnected. What's that? All right. So this whole chapter is very, very different than anything we've done before because we're talking about nitrogen. And the problem with nitrogen in and of itself is that we have no way to store it. Like, we can store carbon in lots of different ways, right? We store carbon as carbohydrates. We store carbon as fats, right? Even though we call it a fat, it's not a carb, right? It doesn't have the C, but it's still carbons. You actually have more energy in fats, but those are all carbon containing substances. So what we have to worry about now is nitrogen, and nitrogen's a little bit tricky. And so you'll see that we'll have very different methods, right? So there are 20 total amino acids, right? We can only make 10, which makes the other 10 essential amino acids. So humans can only make 10. How about bacteria? They make it all. How about plants? So plant, P-L-A-N-T, plant and bacteria, all 20. This is why protein must be a component of our diet, because it contains the nitrogen that we need in order to make all kinds of amino acids. Okay, so if we look, right, at our energy conversion pathways, I kind of like that they always do this. We have a really big integration of nitrogen containing compounds, so amino acids, into all the different pathways we've already talked about. So we can shuttle it off to the citrate cycle, right? We can shuttle off amino acids and convert it to acetyl-CoA. And then once we get acetyl-CoA, what can we go and make? Basically anything. We can go make fats, right? We can make ketone bodies. We can convert our amino acids to pyruvate. We can go up gluconeogenesis, right? So it shares a lot of intermediates. So many shared intermediates. But the source of nitrogen, right? When we talk about what other things need nitrogen, right? Like, do we need nitrogen in acetyl-CoA? No. What else do we need nitrogen for other than amino acids and proteins? Nucleotides. So our amino acids are also going to be the source material for our nucleotide bases. So, and we'll get deep into that. All right, so nitrogen fixation, right? Nitrogen is technically, you know, we say we breathe oxygen, which is true, but really, what's the most abundant element in the air? Nitrogen at like 78%. Like that's huge, right? 70 is 78? 70,
Yeah,
upper 70s.
Less than 1% of other gases and carbon.
Yeah. So we need nitrogen in a lot of different things. We need nitrogen in amino acids. We need nitrogen in our nucleotides. We need nitrogen in some of our cofactors, right? NAD+, FAD, right? There are a lot of different places within metabolism that we need nitrogen. So we have to have a way to pull nitrogen out of the atmosphere and make it usable. So that making it usable begins with nitrogen fixation. So you start out with nitrogen gas. And what is nitrogen gas? What's the structure of nitrogen gas? How many bonds? 3 bonds, right? That's nitrogen gas. We're going to convert nitrogen gas to, what is NH3? But what do we, yeah, but not the structure, but what do we call it? Ammonia. And then NH4 plus is the ammonium ion. So you'll see those, you'll see it switch back and forth quite often between ammonia and ammonia, depending on what's happening, basically. Okay, so you have to reduce the nitrogen gas, then you can oxidize into our nitrate and nitrite, right? And this happens in 2 different ways. Primarily, it happens in bacteria. So in bacteria have an enzyme called nitrogenase. That's in bacteria. It's a huge multi-subunit. We're going to talk quite a bit about it. And then in industry, we use what's called the Haber process. And so this one is basically used in industry. And it's super inefficient. And if we could create a more efficient way to create to do nitrogen fixation, we could feed a ton of people. But we don't know how to do it very well, really. And then a little bit of it happens in lightning, which is kind of cool, but not a whole lot. Doesn't really matter. Okay, so once we've fixed the nitrogen, then we can assimilate the nitrogen, which means that we put it into amino acids. Then it's something that the body can metabolize, the body can use. So if you look at what happens, we're really going to have two nitrogen carrying amino acids. Okay, so we have glutamate and we have glutamine. What's the difference between glutamate and glutamine? How can I convert from glutamate to glutamine? Anybody? You add ammonia. You add a nitrogen group. So depends on if it's NH4 plus or NH3, doesn't really matter, however you want to say that. Okay, so how do we make glutamate? Glutamate's really, really similar in structure to a substrate that we've talked about a lot in glycolysis and citrate and all that. Alpha keto glutarate. How do we go from alpha ketoglutarate to glutamate? It's another NH3. It's just another NH3. So these are two really big nitrogen carrying molecules. That's what we call them. Okay. All right. And so it happens in both plants and bacteria. Does it happen in us? No. So we need to eat them. And it's used by the glutamine synthetase enzyme. And we're going to talk about all the enzymes and how they work and all that kind of stuff. So brace yourself, it's going to get complicated. All right. So we said fixation, right? Fixation can occur in one of two ways. Either it's biological, and biological happens in a bacteria, right? Also algae and that kind of thing, but generally we just kind of say bacteria. And it uses the enzyme nitrogenase. So in order to do this, you are starting with nitrogen gas, right? N2 is a triple bond. Is that easy to do or is that hard to do? That is relatively difficult to do. It takes about 930 kilojoules per mole. I'm going to put a couple of exclamation points behind it just so that we understand that that's a lot of energy. That's a lot of energy. So it requires multiple steps, and not surprisingly, it's ATP dependent because we have to couple it to something in order to drive the reaction. So you're going to see, if we kind of zoom in, right? We're not going to talk, we'll talk about it kind of in general, but we'll go into the details on how this actually works in a little bit. But just so that you know, one's a bacterial enzyme that does the whole process. And then if we want to do it in industry, so we take nitrogen from the air, and then we take hydrogen gas that was derived from methane, and we combine the two under really high pressure, under really high temperature. So 500 degrees Celsius, 900 something degrees Fahrenheit, about maybe 950-ish degrees Fahrenheit. Somebody could do the conversion if they want. 350 kilopascals. We're at about 1 ATM. It's about 3.5 ATM, right? So pretty hot and pretty high pressure. 932. There you go. Okay, I was a little high.
Closer than I was.
And you need a catalyst in order for this to happen. But look at this. We only have a 15% efficiency rate here.
All that heat, all that pressure.
All that heat, all that pressure with a catalyst, and we only get 15% efficiency. if that reaction can change, you double that, you get a Nobel Prize. I'm telling you. So like, if you go on the chemistry route, wouldn't be a bad.
Double efficiency. That's basically what you'd be able to.
Yeah, you'd be a happy person. So you get from that process liquid, basically liquid nitrogen, right? Liquid ammonia. So we can use that for all kinds of different things. The one that we primarily think about is fertilizers, right? We think about that for the fields and that kind of thing. But that's not the only thing. You can make many different chemicals and plastics and fibers and all kinds of different things with that nitrogen. And then also some drugs can use it, which you don't need a whole lot of, right? But really, where do you need a ton of it? You need it in agriculture. You need a ton of it in agriculture, and we'll actually talk about why. Okay, so atmospheric fixation's a little bit different, right? Doesn't happen very often. It's about maybe like 10% of the nitrogen in the atmosphere will actually get converted using lightning. But the lightning is high energy and will again break that triple bond. And then once that triple bond is broken, it can react with the oxygen in the atmosphere. We can make nitrogen oxides. The nitrogen oxides are soluble in water. So they'll solubilize in the rain, rain falls to the ground. Now we have nitrogen in the soil. But it's not a whole lot of it, right? Okay, so this is kind of just to keep a big picture of what's going on in the nitrogen cycle. So if we kind of zoom in, right? How many different types of fixation have we talked about so far? 3. Which 3?
Biological, industrial,
And atmospheric. There's our 3 types of fixation. So regardless of which type you are using, you are going to put usable nitrogen into the soil, right? So either you do it in bacteria, you're doing it in the lab, and then you're going to spread it on the ground, or you're going to do it by lightning, and then it'll fall to the ground as oxides. Doesn't matter. But basically, then you have usable nitrogen that can be metabolized within bacteria, right? The bacteria can incorporate it. The plants can take it up. Some of the legumes, right? You know this, right? You're supposed to rotate your fields. You've heard that before. Why do you always want to rotate your field with legumes? To restore the nitrogen, because those particular plants can do nitrogen fixation, right? So it's a way to basically rejuvenate your soil. So it's actually really, it's got like science background, right? All right. And so we're going to talk about kind of basically a lot of this, where plants will assimilate our nitrogen into glutamine and glutamate, and then we're going to eat it and we're going to metabolize it and all that kind of stuff. Okay. All right. So nitrogen assimilation. If we're talking nitrogen assimilation, what do we want to do? We want to put our nitrogen NH3, NH4+, whatever you want to talk about, and we're going to put that into either glutamate, glutamine. So ammonia and ammonium, either way you want to think about it, that's our nitrogen source. There are 3 enzymes that we're going to talk about in this process. Not all 3 enzymes are found in the same organisms. Glutamine synthetase is in all organisms. Glutamate synthase is found in plants, bacteria, and actually a few insects, which is kind of cool, but we don't generally put them. Then the last one, glutamate dehydrogenase. Glutamate dehydrogenase is a little weird. It is in all organisms, but it's only active when your nitrogen levels are like really high, really high. So think about plants. When would you have a really high level of nitrogen for plants? Fertilizer. So this is when you fertilize plants. UTAL, fertilize plants. Okay. All right. So we need to talk about glutamate and glutamine, right? So glutamate, if we have an amino group that's on glutamate and we transfer it to alpha keto acids, right? We're going to use what are called aminotransferase enzymes. And what we're going to be able to do is to shuttle the nitrogen containing group from one amino acid to another. We'll be able to basically interconvert between amino acids. So you could really say that this one provides the nitrogen for the amino acid biosynthesis, right? It's like the original nitrogen source. Glutamine is going to have multiple nitrogens. And so we call this the primary source of the amino groups to make amino acids, nucleotides, carbonyl phosphate. We're going to go on and on and on and on. But it's a little bit confusing. So when we talk about enzymes, make sure you know, are you hearing glutamate synthase? You're hearing glutamine, right? Glutamine versus glutamate. So just make sure you're paying attention to that. All right. I saw this morning, and I actually complained to the publisher about this. So this slide is about glutamine synthetase. What reaction do you see in that picture? Glutamate synthase. So is that the right reaction? That ain't the right reaction. I complained to them about this specific thing, and they didn't update it in the new edition. But anyway, okay. So we are going to talk about glutamine synthetase, but I think it's really good. It's actually, we're gonna use this as a plus because we're going to start drawing some stuff, and I think that's going to really help solidify it for you. So what's the difference between synthetase and synthase? Remember we've talked about in the past? Synthetase requires ATP. So you look at the reaction there, it doesn't require ATP, so it's a synthase, right? But you're still creating something. All right. So the reaction we're going to talk about, it's a synthetase, so it requires ATP. And we're going to use our ammonia, our ammonium ion, and we're going to convert glutamate to glutamine. So I want to draw this. Y'all can do this. We're going to draw this. We're going to draw this. Okay, so glutamate, U-T-A-M, glutamate. Don't look at the screen. Don't look at the screen. How do we draw glutamine? Glutamine, remember glutamine and glutamate. This is how I remember. Glutamate wants a mate. So there's only one nitrogen because it wants a mate. Glutamine has its mate. So it has two. Okay. So basically the structure is the same otherwise. So you have, right, a carboxylate ion, right, that's attached to a 3-carbon long chain. All right. One. All right, a 3-carbon long chain. So we got 1, 2, 3 carbons. Then we're going to end in another carboxylate. You can put the C there if you want. All right, it's got 3 carbons in the middle. You have 2 carboxylate ions and 3 carbons in the middle. That's your common, basically your common structure. So if we look at glutamate, glutamate has a nitrogen right here. It's the charged. Be careful, it is charged. So this is really a 2-step reaction mechanism that requires ATP. So the first step, it's still the same, it's the same enzyme that catalyzes this, but it's a 2-step reaction. So our first step is what requires ATP, and we're going to generate ADP, right? So what we have to do is we have to prime, you have to prime this molecule to accept a nitrogen. So a lot of times, how do we do that if you know you're using ATP? what is your intermediate going to look like? It's going to be what? It's going to be phosphorylated, right? It's going to be a phosphate group because we're going to take the phosphate off of ATP and put it onto glutamate. So this is glutamate. So I'm just going to copy it. I'm going to cheat. Right?
That's not fair.
I know. Okay, so I'm going to cheat. And right here, put a different color. I'm going to put the phosphate group. I'm not worried about the details.
So this is glutamate, right?
This is glutamate.
So if you remove the.
I'm going to make glutamine. I'm making glutamine. This is glutamine synthetase, which is going to make glutamine from glutamate. Yeah. You got to be careful on this one. Okay. So now, look, I'm going to cheat again. Yes, I'm going to cheat again. I'm going to cheat again. Okay. So, so what's going to be different? The only thing that's going to be different is the site where I primed my molecule. I'm going to replace that with a nitrogen containing group, but this one is uncharged. So I started with glutamate. This intermediate is alpha glutamyl glutamyl phosphate. I just can't write and talk at the same time. I have this problem. That's gamma. I'm sorry. I said alpha. It's gamma. It's gamma. glutamyl. Glutamyl. Okay, so when I went from my intermediate to glutamine, what do I have to release?
The phosphate group.
I have to release the phosphate group. And what do I need? I need a nitrogen group. So see how related they are? Glutamate is waiting for its mate. Glutamine has it. What?
What does it say? NH4 plus?
Yes, NH4 plus. Here? Yes, NH4 plus. Okay, so yes, it's the same enzyme. It's a two-step, same enzyme. This is all done by glutamine synthetase. Same enzyme. It's just a two-step mechanism. Okay, so I want to talk. So are we okay on structure? We okay on this? Maybe. Okay, so I want to talk glutamine and glutamate synthetase. So if I don't want to draw structure, if I just want to draw names, right? I'm going to start with glutamate. I'm going to add basically overall, what do I need overall for this reaction to happen? Like, yes, you need ATP to drive it, but this is an assimilation, right? So what am I assimilating? Nitrogen. So I need a nitrogen group, right? And if you want to put in your ATP, you could put in your ATP like that. So this is my glutamine synthetase, and I'm going to make glutamine. But be careful, I don't want you to get mixed up. We also can convert glutamine using, in addition, alpha keto glutarate, but it does not require ATP. It requires, we're actually reducing it. So we require a reducing agent here. So we're actually going to use NAD or NADPH, right? One of the two. And we're going to make, I can move it up, glutamate.
So they can kind of each other.
Yes, but it's different. So this is glutamate synthase, syntase, synthase. This one is glutamine synthetase. So be careful with that. Be very, very, very careful with that. So glutamine synthetase is really important, not only as a primary entry, right? This one right here is our primary entry, for nitrogen into basically biomolecules, but it's also really, really important in animal cells as a means to transport nitrogen, because we have to be able to get nitrogen from one place in the body to another. And so this is going to be the way that we carry it. And we'll see that much later. So far, so good? Primary entry for nitrogen in biomolecules, but also for transport of nitrogen in animals. I'm so proud of y'all for reading my handwriting.
Is it always going to be NH1 plus, or is it going to be switched with NH3?
It
Doesn't matter.
doesn't matter. It doesn't matter.
What's getting retained is the nitrogen.
Yeah.
Okay.
It doesn't matter. Depends on what the original source was, but it doesn't really matter.
Either way, we're just looking at nitrogen, not the protons.
Correct. Well, you take some of them, but
But
they're not the important part. They're not the important bits. They're not the important bits. Okay. Now, oh, did we cover everything on here? Yeah, okay, so we did talk about this, right? Requires ATP. And then this is how we said it's important for integration of our ammonia, right? From glutamate to glutamine. So it creates glutamine. It's a way to assimilate that nitrogen. Then we said it's also important in animals because it transports nitrogen from peripheral tissues to the liver. So that's everything we said. Sometimes I get to chit chat. I forget. Okay. So hugely important enzyme. So what do we have to talk about? Regulation. We have to talk about regulation. In bacteria, this is a massively complex enzyme. It's got 12 different subunits. So it's really big. But you need to have ways to really, really tightly control this. So of course, you're going to do it two different ways. What two ways are you going to control any enzyme? Inhibition. So feedback inhibition is allosteric, right? And covalent modification in this case is going to be through adenylation. So our feedback inhibition. Remember the pathway, we're going to make glutamate, we have glutamate, we're going to make glutamine, right? We incorporate our nitrogen. That glutamine is then going to go on and be used to make lots of different things. Those things that are final products can come back and inhibit through feedback inhibition allosteric the activity of the enzyme. Is that what we want? Absolutely, absolutely, that's what we want, right? What else is going to inhibit it? Alanine, glycine, serine, all what? All amino acids. So if you have a ton of amino acids already floating around, do you really need to make more glutamine? Not really, you don't. So those are also going to be inhibitors of the process. So you have lots of allosteric inhibitors. We have one covalent modification. So let's talk about the covalent modification, right? Oh, in order for this to happen, what three things do we need to have bound, right? what three things, if you have the glutamate synthetase enzyme, what three things do you need? Three things need to be bound. What three things? ATP, a nitrogen group, and glutamate. Glutamate. Glutamate is the substrate for glutamine synthetase. And all of these inhibitors down here, these are all synergistic. So the more different things you have, the more you're going to inhibit it, right? So you could write that synergistic. Synergistic. So there's additive and then there's synergy, right? So if you have something that is additive, if I inhibit the enzyme by 10% and you inhibit the enzyme by 10%, if we are together, we inhibit the enzyme by 20%. If you and I are synergistic, I inhibit by 10%, you inhibit by 10%, together we inhibit by 90%. Or some greater number than the additive. It depends on what it is, what that degree is.
So synergistic just means that working together, there's a lot more
Correct.
efficiency with it.
Correct. Exactly. Okay. All right. So remember, adenylation, if we're talking adenylation, we're doing what kind of modification? Covalent. Okay. So this little purple thing, right, is our enzyme. This is glutamine synthetase adenyltransferase. So this will adenylate our glutamine synthetase. So this green is glutamine synthetase. This is the inactive form. This is the active form. If it is adenylated, if it has AMP, is it active or inactive? It is inactive. If we have something where it has been de-adenylated, active or inactive, it is now active and it can convert glutamate to glutamine. Now, the enzyme that adenylates it is also covalently regulated. So this one enzyme can both adenylate and make it inactive, and the same enzyme can de-adenylate and make it active. So how do you have one enzyme that does both things to the same substrate? You have a regulatory molecule on the end of it, right? So this is the P2 domain. If the P2 domain has, what is UMP? Monophosphate, right? So remember how we've done that in the past multiple times now, where we have monophosphate of whatever it is, and it regulates. So here, if we have on tyrosine 51 UMP present, then this enzyme is active. If the adenyltransferase enzyme is active, it is going to de-adenylate and make our glutamine synthetase active. Whew, right? Okay. So now, what if you have this enzyme and our adenyltransferase enzyme does not have a UMP attached to it? It is active. Because it is active, it can actually adenylate our glutamine synthetase. If our glutamine synthetase is adenylated, it is inactive. Yeah? Yes. Not whether it's inactive or it's active. It's whether it is adenylating or de-adenylating glutamine synthetase. Okay, so if you do not have UMP, it is going to adenylate. So no UMP on the adenyltransferase, then we're going to put AMP on glutamine synthetase. if we have AMP, if it is adenylated, it is inactive. If the regulatory enzyme, the adenyltransferase, has a UMP present, then it is going to de-adenylate glutamine synthetase. It's a little bit like those phosphorylation cycles, right? But this enzyme just doesn't have an on-off switch. This enzyme has an adenylate de-adenylate switch. It's always active. It's either adenylating or it's de-adenylating. It doesn't have an off switch. But if it de-adenylates, it creates an active glutamine synthetase. Okay, good. All right. Do you have to know this? Yes. This next part, like you don't have to know as much as you think you're going to have to. Okay. So how can we transfer these nitrogen groups? Well, y'all have already told me that alpha ketoglutarate is a great acceptor molecule, right? We've already talked about that. So I can have any alpha amino acid in combination with alpha ketoglutarate, and I can make an alpha keto acid and glutamate. Because what did I do? I transferred the nitrogen group from my amino acid, and I hooked it on to alpha ketoglutarate. Now I have glutamate and an alpha keto acid, right? So this happens through the activity of an aminotransferase. Because what are you transferring? The amino group, right? The amino group. You're transferring the nitrogen. Most of these reactions are completely reversible, but it could be any amino acid, right? You have specific enzymes that are specific for the different amino acids. We have most of them, not all of them, but we have most of them. It requires a cofactor, PLP, and PLP is actually from vitamin B6. So it is something, right? You remember how we talked about in Biochem 1 that lots of cofactors and things like that were vitamins. And why are they vitamins? Because we can't make them ourselves, but we need them in chemical reactions. Same thing here. Same thing here. Okay. So the alpha ketoglutarate is what's serving here as our acceptor molecule, right? So we're going to look at the mechanism for this. Do you have to memorize the mechanism? No, you do not have to memorize the mechanism. I want you to understand, but I do not want you to memorize this mechanism. We learned mechanisms so that you understand how they work, but we are not going to memorize them. Okay? But it's really important that you remember we start out with an amino acid and alpha ketoglutarate. What do we end up with? An alpha keto acid and glutamate. So glutamate. Okay.
Sorry.
No, that's okay. That's okay. So here's our mechanism, right? So this is really, it's like we're going to play ping pong with the nitrogen group. That's kind of basically what happens. So in stage 1, right? This is separated into the blue and the green. The blue is stage 1, the green is stage 2. So if you look just at stage 1, right? So what we want to happen is we want to move that alpha amino group, right? So kind of keep in your mind what's supposed to be happening. The alpha amino group is going to be transferred to what? Eventually. To the acceptor molecule, we want it to go to alpha ketoglutarate, but we're going to have intermediates, right? So we're going to take that alpha amino group and we're going to transfer it to the enzyme bound PLP, right? That's our cofactor. And once we make that transfer happen, then what we're going to do is we're going to form right down here. This is the end of stage 1. We're going to form pyridoxamine phosphate, and we're going to form an alpha keto acid. So all this happens through the formation of Schiff bases. Did y'all study those in? Some yes, some no? Yes, no? So just what we're going to do is we're going to incorporate the nitrogen into basically into a ring, right? So if you look first at your Schiff base, right? We have our nitrogen in the ring, and then here's the enzyme. The enzyme has a lysine residue, right, that has the nitrogen. So what happens when the amino acid comes in is the nitrogen that was here is going to be replaced by the nitrogen from the amino acid. So the nitrogen that was in the ring is now separate and attached to the lysine residue of the enzyme. The new nitrogen that came from the amino acid is now present in the ring. Okay, keep going. And what we're going to do is we want to keep the nitrogen, and we want to kick out the carbon containing molecules, right? So as we go down here, we add water, we can release the alpha keto acid, right? That was one of our products. Okay. We can bring in a different alpha keto acid, right? So now we have this nitrogen was from the original amino acid. These carbons are from a new amino acid. So you can have a different R group here than you had in the blue, right? So we have our second amino alpha keto acid that comes in so that we can make a new amino acid. So the whole point of this is to be able to switch between different amino acids, right? Okay, so now we're basically building the structure for an amino acid. What do we want? NCC, right? That's the backbone of an amino acid. So that's exactly what we want, but we don't want a double bond here, and we need to free this nitrogen. So we lose the double bond. Now this nitrogen is going to take the place of the blue nitrogen, and we've regenerated our enzyme, and we create an amino acid, basically where it's basically a new amino acid. We transferred the nitrogen from one amino acid to another. Yeah? Do you have to memorize the mechanism? Sure. No. Sure. I'm not doing that. I'm not doing that to you. I'm not doing that to you. So most animals have like 18 different aminotransferase enzymes. We don't have all of them, but we have most of them. We're missing lysine and threonine. I believe it's threonine. I have to go back and double check. I know it's lysine. And the reactions are reversible. All of these reactions. So do you see whenever we see any kind of, whether it's aspartate, it's alanine aminotransferase, do you see how they're double-sided arrows? They're reversible reactions, and they're reversible based on the level of the different amino acids present in the cell, right? So if I'm creating a protein that's really rich in alanine, I'm going to deplete my alanine, and I'm going to need to shift to make more alanine, right? And vice versa. So this is a way for the cell, because it's reversible, that dependent upon the cellular conditions, you can just generate whatever it is you need. Yeah, it's feedback. It's feedback, right? You lose a product, so you go make more, right? Okay, so basically it's interconversion of amino acid pools that reflect the protein synthesis needs of the cell. So let's take these two reactions in general, right? We're converting aspartate to glutamate. Why do I want to go from aspartate to glutamate? If I need alanine, how can I make alanine? If I start with aspartate, I have aspartate, and I want to make alanine. That's right. Okay, so this is overall what I want to do, but I can't go straight there, right? I'm gonna have to combine these two reactions to make one overall reaction. I'm gonna couple them, right? So the first reaction, I take aspartate and I'm adding what to it? Alpha keto glutarate, right? When I add alpha keto glutarate, it's reversible. I make oxaloacetate, OXLO. I get it so ahead of myself. Glutamate. Okay, so now I can basically flip-flop the bottom reaction, right? So I have glutamate plus pyruvate, and it's reversible to make glutarate plus alanine. So then what ends up happening, right, is you eliminate your intermediates, and I'm able to convert aspartate to alanine. So you have different, these are different aminotransferases, right? The bottom one's alanine aminotransferase, the top one is aspartate aminotransferase. There's one for every amino acid. Yeah, I'm sorry. They switched the other two. Yeah. We're gonna fix it. This is why I was so mad. So before we talk, y'all got that? Yeah. Okay. So this says it's an indicator of liver health. Why is it an indicator of liver health? Well, we normally find it in muscle and liver. We find these enzymes, VER, liver, but not normally in your blood, right? So this is very similar to a heart attack, right? If your heart cells die, then heart proteins are found in, heart enzymes are found in the bloodstream, right? So if you have liver damage, liver cirrhosis, hepatitis C viral infections, right? All that kind of stuff. Then your cells are going to die. They're going to lyse open. They're going to release these aminotransferase enzymes into the bloodstream. So they're an indicator of liver health. Kind of interesting, right? Okay. So now we have to be unhappy with well, not so much. Okay, glutamate synthase. I wanted to say synthetase, but it's not, it's just synthase. So glutamate synthase is a reductive amination of alpha ketoglutarate. So if we look at our glutamine synthetase, right? Remember, in our glutamine synthetase, we take glutamate and we make glutamine, right? So now, when we look at glutamate synthase, we take that glutamine, add alpha ketoglutarate, and we go through what kind of a reaction? Oh no, this is why you don't gesture with this pen. I lost the top. I'm not gonna be able to write. Where'd it go? Y'all might have to turn on the light for a second. Turn on the light. Yeah. Do you see it? I heard it hit the ground and drop. Oh, y'all have so much better eyes than I do. Thank you so much. What's that? Bonus point. The worst. I know. You can't see that. Okay. Yeah, I have a pen. It's got the tip.
Oh, you lost the bit. Oh.
Yeah, I lost the very tip. You can't. You
For some
can't.
reason, I was just like, it doesn't matter. Just give it after class, but then I realized.
No, it's not the back end.
Well,
It's
no, I
the
thought it
tip.
was like a regular pen with like
Oh,
a cap
oh,
on it. So I was like, it doesn't matter. Just don't write on the board.
Okay. So glutamine, right? Glutamine has how many nitrogen groups? 2. So if we have something that contains, we're going to put 2 in, right? How many nitrogens in alpha ketoglutarate?
Oh wait, 0, sorry.
0,
My brain's
0.
in my head.
right? Now we're going to make 2 glutamates. How many nitrogens in glutamate? They want to mate, so they only have 1 in each. So we can convert glutamine to 2 glutamates. So why is this reaction really important? So the whole point, what we want to be able to do is we want to be able to create nitrogen carrying molecules. So is alpha ketoglutarate a nitrogen containing molecule? No. Then we have our nitrogen, and from that we want to make glutamate. So it's really a combination of these 2 reactions that allow us to convert alpha ketoglutarate to glutamate, right? So you go from having something that has no nitrogen containing groups, add a nitrogen group, you're making something that now contains 1.
So we have to start off with glutamate.
You have to have glutamine to make more glutamate.
But in order to make glutamine, you have to have glutamate.
Yes. You have to have a stockpile. You have to have some. Like you have to have some. You eat it. It's cool. You eat it. You have it. Well, we're not trying to make it from scratch. We're just trying to transfer nitrogens. You gotta remember that this is a net reaction. We don't integrate nitrogen containing substances like ammonia and ammonium ion. We can't do that. What does that? Bacteria plants, right? All we want to be able to do is to transfer the nitrogen group. So when you look at a net reaction like this, you're thinking, oh, we can assimilate nitrogen. No, we can't. No, we can't, right? What we're just doing is basically shuffling around the nitrogen. Okay?
So, do you get nitrogen, like the nitrogen groups because we eat them?
Yeah, we eat them. No. You can inhale it, but you're not going to assimilate it. No, you're not. No, that would be really cool, but no, we cannot do that. That would be awesome.
Imagine being like, what's your favorite fat? Oh, I can inhale and
I can assimilate
I can
nitrogen.
assimilate my own nitrogen.
Yeah, no, you can't.
Share.
No, you can't.
No, that is.
Yeah, we can't do that. We cannot do that. Plants can. Plants, bacteria, right? Okay. Glutamate dehydrogenase. This is an enzyme that we all have, right? So this one interconverts our alpha ketoglutarate to glutamate in the presence of NH4. So this is only functional when NH4 levels are super high. So what was that condition that we talked about? The fertilizer, right? If we put a ton of fertilizer on the ground for those plants, then they're going to create glutamate from alpha ketoglutarate, but only in the presence of a ton of nitrogen. Why? Look at this reaction.
It's not energetic favorable.
It's a plus 30. It's a plus 30.
It's gonna be hard.
That's a lot.
That's a lot. It's a lot. So which enzyme do we not have? Glutamate dehydrogenase, glutamate synthase, or glutamine synthetase? Which one do we not have? We do not have glutamate synthase. So humans, no for humans. No for humans.
That's bacteria.
Bacteria, plants, right? Yep. Mhm. But we don't really do this because it's energetically unfavorable.
We do have it.
Technically, we do have it. Technically. Okay. So that is perfect end. Yes, we have time. Woohoo. Okay. So glutamine synthetase is super complicated, right? super, super complicated. So it's important when you're thinking about these enzymes to kind of organize all the information about them in mind. So there are a few things on this one that I want you to try to answer on your own or you get into a group. And then I am going to go and make this Jamboard. Now, have y'all ever done a Jamboard before? Okay, well, just do your little thing and then then you're gonna group together and then we'll create something. Okay, I'm gonna end the Zoom because y'all are just gonna be working.